The overall goal of the following experiment is to capture the real-time dynamic process of phagosome maturation and phagolysosome biogenesis in phagocytes. This is achieved by loading the lysosomal compartments of the cells with fluorescent conjugated dextran to enable the visualization of lysosomal compartments and transfer of its luminal cargo to phagosomes. As a second step, microparticles are added which act as a phagocytic model. Next, the delivery of lysosomal material to the phagosomes is analyzed using live cell imaging and subsequent digital image processing in order to quantify the phagosome maturation process. The results show the effect of various factors on the process of phagosome maturation based on the delivery of lysosomal content to the phagosomes.
The main advantage of this technique over existing methods, such as the evaluation of macrophage maturation in fixed cells, is the quantitative readout with a high temporal resolution.
Begin by dissolving Texas Red labeled 70 kilodalton dextran, or Dex 70 Kd, in PBS at 20 milligrams per milliliter and store aliquots at -20 degrees Celsius. Next, dilute the Dex 70 Kd stock in complete cell culture Dobecco's modified Eagles medium, or DMEM, to approximately 1 milligram per milliliter. Sonicate the dextran aliquot in an ultrasonic water bath for 5 minutes. Then, centrifuge the solution in a microcentrifuge at maximum speed for 5 minutes to remove any clumps or contaminants from the solution.